Good evening. Oh, should have waited for that.
Yeah.
I hope everyone's good with this being recorded today, but just letting you know for privacy that we are recording this meeting. My name's Claire Bailey. I'm one of the founders and directors of Duchenne Australia. I do know most of you on the call, but those that I haven't met before, welcome and thank you very much for joining us tonight. I'm just going to do some quick introductions. Introduction to Michael from our team. Michael, do you want to do that, or do you want me to?
Yeah, you can go for it, right?
So Michael Simpson, also known to a lot of the families online. Michael is also co-founder with myself of Duchenne Australia and director. He has a 10-year-old son, Harry Harrison, with Duchenne and lives in Perth, which is where I'm based as well, over here in obviously Western Australia. And I have a son who's turning 22 tomorrow with Duchenne. So Michael's got 3 children. Harry's his eldest and is the only child with Duchenne. And I've got 3 children, and Logan's the only one with Duchenne as well. So thank you for joining us today. I'd like to acknowledge the traditional owners of the lands on which we're meeting today. So wherever you are, and pay our respects to elders past, present and emerging. And today's session is a really exciting opportunity for Duchenne Australia, but also all of our Duchenne families and all other stakeholders across our networks to understand a little bit more about the exciting research that's being undertaken at the Murdoch Children's Research Institute. But also the important role that data and registries play in that. So as much as we get excited about emerging therapies and research, we can't do any of that without understanding the community and, you know, having access to all of that really valuable and rich information. So it's we're going to be introducing our first speaker, Robin Forbes, very shortly, who plays the pivotal role in the Australian Neuromuscular Disease Registry, which is also based at the MCRI. So without further ado, I'll introduce Robin Forbes, who's a genetic counsellor who's worked in the multidisciplinary neuromuscular clinic at the Royal Children's Hospital in Melbourne since it first began, which is back in 2008. She has recently changed roles, and Robin might give you a little bit of overview of that in a second. But the clinic oversees the care of more than 400 children with a variety of neuromuscular disorders, and that's right through all the genetic sort of counselling and support that she provides to families, but right through that whole time that families spend at the MCRI in the paediatric services. So Robert will be overseeing the day-to-day running of the Australasian Neuromuscular Disease Registry, but she also plays a very, very important role in the data collection, but also helping families understand what their child's genetic diagnosis means, and then what potential therapies obviously coming through would be their child may be eligible for. So Robin, I'm going to hand over to you.
Great. Thank you, Claire, and thank you, Duchenne Australia, for asking us to speak this evening. And I guess I want to make one point. So Peter Howling, who is one of the senior scientists at the Murdoch Children's Research Institute, we both are based at the Royal Children's Hospital in Melbourne. And even though it's the Royal Children's Hospital and the Murdoch Children's Research Institute, we are both very much involved in national, so Australia-wide, care and data and research for adults and children with neuromuscular conditions. So just because we're based in a paediatric setting, everything that we're saying tonight is about adults as well. So I just want to preface that while I think of it. So if you're a young adult or an adult, or you know an adult with a neuromuscular condition and you're listening in tonight, this is relevant for you as well. So I just wanted to make that point. Next slide, please, Pete. So I would like to acknowledge, in the spirit of reconciliation, that the ANMDR and the Murdoch Children's Research Institute acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respects to their elders, past and present, and extend that respect to all Aboriginal and Torres Strait Islanders people today. So as Claire mentioned, I'll be talking to you this evening a little bit about the Australian Neuromuscular Disease Registry and more specifically the Duchenne Registry, which is an important part and was the first part of that registry, and why we are collecting the data very briefly, some of the data that we are collecting, and then how that evolves with research and, what are we doing with this data that we're collecting. So the registry, as we said, is based at the Children's Hospital in Melbourne at the Murdoch Institute. And we have an advisory committee that oversees what we do. Patients can self-refer to the registry via our webpage, and there'll be a link to that at the end, or their clinicians might tell them about the registry. Importantly, the registry is for children and adults across Australia with neuromuscular conditions. And we've listed there the conditions that we are currently collecting information for. And we have ethics approval to do this, to have this data collection. So we're not just collecting data willy-nilly, what we feel like doing on a whim, but this is has ethics oversight, which is incredibly important for anything that you're thinking of being involved with or thinking of doing. Always check that there is ethics approval, that there is oversight to what you're potentially thinking about being a part of. The registry, I think very importantly, is a part of the Treat NMD Alliance, and this is based in the UK. And this is a global network of national patient registries for patients with neuromuscular disorders. And Australia has very quickly played a very important role with Treat NMD, and we were the first country to roll out the new Duchenne data set. And we also were the first country to roll out the limb-girdle muscular dystrophy data set. So Australia, with its research record, is also leading the way with Treat NMD. Being a part of that registry means that we're asking the same questions in Australia as what they're asking in Canada, as what they're asking in the UK. So it means that we can get information and data very quickly, not just on Australian patients, but on patients around the world. Information is kept confidential, so everything is de-identified on the registry, but it's a way of answering questions quickly, and particularly when time is the enemy. Treat NMD has about 60 countries involved now on their registry, and they have over 80,000 patients on their registry. So that's a massive number. Next slide, please, Pete. So this is just a quick snapshot. We now have almost 800 people on the Australian registry. And initially, we just started collecting information on 4 major neuromuscular conditions. And then in the middle of last year, we added some rarer conditions, because we know that disease-modifying therapies and different treatments and research is coming on board. So it's important that we are collecting the information on these other rarer conditions as well, so that we can learn about the natural history and get the natural history data for these neuromuscular conditions. And what natural history means is what happens with this condition when there is no treatment available. Next slide, please, Pete. So the Duchenne Registry, I will talk about now, and that has 112 data items. So if you're on the registry, you'll know that our team often ask you many questions about what the outcome measures were from your last physiotherapy appointment, or what your last lung function test was, or your cardiac review. But it's important that we have the most up-to-date and accurate information that we can on either you or your child. And it's important that this is robust longitudinal data, that we're collecting this information over time. And the data set was put together by under the guidance of world leading experts in Duchenne muscular dystrophy. So we know that the data that we're collecting is relevant to the Duchenne community. And then registry staff contact you and update that information annually. Thanks, Pete. So why an Australian registry? And this is incredibly important. And I think first and foremost is we want to help facilitate the best practice care and service provision within a national context. So many of you will know that there are standards of care that have been published in the medical literature, that it's available online. for the standards of care, or what the care that we should be offering for patients with Duchenne muscular dystrophy. So by having this data on the registry, are we meeting the standards of care in Australia? And the answer to that is once paediatric patients transition into adult services, we know it's much harder to maintain standards of care. Most states around Australia do not have adult multidisciplinary clinics like we have in the paediatric setting. So the data that we have on the registry, we hope can be used to help improve the care for adults with Duchenne muscular dystrophy. The data also helps us understand the epidemiologic landscape of neuromuscular conditions in Australia. So it helps us understand what's happening with Duchenne in Australia and with other neuromuscular conditions as well. But in rare disease, and believe it or not, Duchenne is considered a rare disease, no one country is enough. So being part of the Treat NMD registry helps us tackle issues collaboratively. And we are working internationally, looking at, for example, I'm on an international working committee because of the registry, looking at adding Duchenne to newborn screening. And if we do that, how that should be implemented and so on. So we're working together with world experts nationally. But importantly, and part of what this talk is about tonight, is that the registry can provide a platform to support and accelerate neuromuscular disease translational research. And that's what Peter will talk about a little bit more later on in the talk tonight. So whilst we have good patient data, why are we collecting that data? Why have all this information if we're not going to do it? We need to translate that into research as well that will help the people that we have on the registry. And when we first set the registry up, which was only in 2019 and the beginning of 2020, it was very much to become trial ready. So we needed to know where patients were, so that when clinical trials come to Australia and we're setting up clinical trials, where are patients around Australia? You don't need to be in Melbourne or Sydney in the big research centres. You know, where are you in Australia? Northern, Western Australia, in the Northern Territory. So the registry helps us know where you are, so that if a clinical trial becomes available that your child may be eligible for, or an adult may be eligible for, we need to know about you. What has happened very rapidly in the last couple of years is now to become treatment ready. Things are changing very quickly in the neuromuscular landscape. And with the advent of disease-modifying therapies, so in another condition called spinal muscular atrophy, or SMA, there are disease-modifying therapies available like gene therapy. The registry is now used as a post-marketing surveillance. So once we start giving people gene therapy for Duchenne or gene therapy for spinal muscular atrophy, does it work as well in the real world as what it did in a clinical trial? And clinical trials are very controlled, very monitored, and in some ways a little bit artificial. But when a drug or a treatment or a therapy is approved, does it work the same way in what we call real-world evidence and real-world data as what it did in a clinical trial? And so the data that we have on the registry is a way of using post-marketing surveillance. And for a treatment or a therapy to be approved, post-marketing surveillance needs to occur. Thanks, Pete. So the data that we have, as I mentioned, is on paediatric and adult patients. And if you're not on the registry, I'd love you to contact us to discuss the registry, to see if you're interested in being on the registry, and we can certainly talk to you about that. To be on the registry, you do need to have a genetically confirmed diagnosis, and I would hope that there are no patients with Duchenne muscular dystrophy that don't have genetic confirmation of their diagnosis. But if you don't, then that's something that we can also talk to you about and perhaps help you facilitate getting that performed locally. And then we update your information annually. The aim of what we're doing, and we keep revisiting this, is that we have harmonisation, not just on our registry, but internationally, with well-defined disease-specific data sets. So the questions that we ask patients with Duchenne on the Duchenne registry are different to the questions that we ask a patient with spinal muscular atrophy, and different again to a patient that may have limb-girdle muscular dystrophy or another condition called FSHD. Because even though all of these are neuromuscular conditions, they are all different. they will all have different therapies and disease-modifying therapies and treatments that become available. So we can't ask the same questions. They are disease-specific data sets and disease-specific questions. And that is how we will get the best information and the quickest answers regarding your particular condition. And the way the data sets are developed is it's using a consensus-based approach that involves key opinion leaders around the world. Registries are involved, we involve clinicians, and we involve patients on, are we asking the right questions? So for example, with spinal muscular atrophy, a patient may be able to move a finger a bit more after they've had therapy, say an adult with a condition that's got treatment late. And someone might think, oh, that's not a big change, but someone who has a powered wheelchair, who can actually move the wheelchair themselves or can actually lift a hairbrush to be able to brush their hair, those are important activities of daily living that we need to be able to measure, but we need to know what questions to ask. So we involve patients as well. And the Australian registry is quite a unique model. Everywhere else around the world is either a patient-based registry where patients enter their own data, or a clinician-based registry where clinicians just enter the data. We have staff that try and do the heavy lifting for the patients. So we chase up information that you don't know. With your permission and your consent, we will try and chase up that information for you. But importantly, we work very, very closely with the researchers in our area. Claire mentioned that I'm a genetic counsellor in the neuromuscular clinic at the Children's Hospital, and I've seen every new family that's ever come through the Children's Hospital since 2008 and involved when the families are given a diagnosis in their child. So for us to be able to do that day after day, which is quite grueling, we need to have to work closely with the researchers who are also based around us, and to have good information like this on the registries, and work closely with the scientists. And I think that is also a unique model, that we are able to work so closely together. This is actually the second talk that Pete's given today. He spoke to the neurologists and the neuromuscular, the whole neurology department at the Children's Hospital today. So, you know, people are keen to hear what is happening in this space. So I want to say, don't lose faith and don't lose hope. So we have this data. What are we doing with it? And this is actually quite exciting. So last year we were part of a Medical Research Futures Fund, which are incredibly hard to get. And this is a national functional diagnostic program for therapy development in congenital muscle disease. This is incredibly exciting. And it's a national collaboration between 14 clinicians, scientists and researchers from all over Australia, and recruiting patients from the registry as well, from other places as well. But that's a nice segue to what Peter's going to talk about, and I'll hand it over to you, Pete.
Great. Thanks, Robin. So, yeah, so I'm a research scientist here at the Murdoch Children Research Institute. And this is, like Robin said, a really great example of how we are working together, both in a research capacity and from a clinical setting, with a nationally funded program, which has got a long title, a long name, the Australian National Functional Diagnostic Program for Therapy Development in Congenital Muscle Diseases. But really, it's a network. So on the right-hand side, we can see this really exciting clinical network that covers everyone from Victoria, New South Wales, Queensland, South Australia and Western Australia, which provides a clinical network. And then we fit in down here looking at some of the cellular disease model platforms and therapeutic screenings. And the idea of this is to really generate patient-led data to be able to identify new treatments and therapies. So this is kind of like a bit of a taster of what we do in the lab. But so where I am, we're in the muscle research team here, and this is some of our team. But my group leader is Professor Catherine North, and she is also the director here at the Murdoch Children's Research Institute. So we're really fortunate to have such great leadership. Some of you may have heard of Kathy before. She's pretty well known in the neuromuscular space. She's a clinician as well as, well, ruler of all things tends to be what comes up with Kathy. But our team, our group, really has quite a broad focus. And this slide is trying to represent what we do within our group. And we look at everything from, on the left-hand side here, some of these mechanistic and preclinical models. So this is before we come to patients, but really trying to understand the causes of diseases. All the way to the other end of the spectrum, where we're working with the clinical guys in the lab to test new treatments and therapies and potentially identify new drugs and targets for these kids with neuromuscular conditions. And in the center, much like what Robin was talking about, is sort of the patients. And in our case, that's looking at biobanking and natural history studies. And I'll come back to this in a little while, but just want to give you like a bit of a flavor of the whole lab and what we're working on. So our muscle disease research team was established in 2018 as part of the MCRI stem cell medicine program. And this is a really great initiative that's been led by Melissa Little, who is another world leader in stem cell research, particularly in kidney disease. It's a strong collaboration with the neurology clinic here. And this was predominantly with Monique Ryan before she turned teal and abandoned us for the political spectrum. And now it's led by Epi Robin and Ian Woodcock. But we also have really strong interactions with patient organizations and groups like yourselves. And this really helps us to drive the research that we do, both in terms of motivation and that kind of communication between researchers. And I just want to touch on 3 kind of core programs that we're doing. And the Neuromuscular Disease Registry, which Robin's just talked about, sits up here. But all of these programs really center around the patient and what we can do with and for patients. And so the first program that I want to talk about is a biobank. And so while the registry is excellent and 100% needed, for us in the lab, what we need is patient material and samples to be able to work with. And so in 2018, we identified when we started up this lab that there was a bit of a lack of samples that had been correctly consented and were available for research for the things that we wanted to do. And so we worked with the Melbourne Children's Heart Tissue Bank, which is based here, and Dr. Adam Pears, who actually did his PhD in our lab. He's a muscle disease researcher who was stolen by the cardiac guys. We still chase him every now and then. But he really helped us to start to collect, particularly blood, from patients with genetically diagnosed muscle disease. And these are adults and children. And this is sort of a list of the conditions that we have here. Duchenne was, again, first and foremost on the list of patients that we were actually consenting. Our first patient into the bank was also a Duchenne patient that Robin had consented. So Robin has been integral in collecting all of these patients with us and giving us the opportunity to work here. In 2021, we started to work with Ian Woodcock and Katie DeVal on really pivotal study looking at natural history of children with early onset FSHD. And then in 2023, we were actually able to take our bank and we're looking at creating this national collaboration to be able to help harmonise what's happening around Australia. And so this we're calling the National Muscle Disease Biobank. And it was set up just this year, and ethics and governance is run through the Royal Children's Hospital. The bank, or at least part of it, is housed here at the Murdoch Children's Research Institute. And biobank is really a collection of both clinical data, like what the registry holds, but also blood and skin fibroblasts when they're available, and occasionally muscle biopsies that have been collected from adults with a diagnosed genetic muscle disease. Now, this bank is sort of disease agnostic. We cover the breadth of conditions that are around Australia, particularly because of the rare, ultra rare and understudied kind of aspects. And like what Robin was saying with the registry, no one country can really cover the breadth of these conditions. And the same holds true for some of these materials. So, you know, the hope is that by developing this bank, we're able to actually collaborate and share the samples and materials that are coming from Australia internationally and both nationally within Australia. So I mentioned this is a national approach. So we have centres that in Queensland, Sydney at the Sydney Children's Hospital Network, here with us at the Royal Children's, the Women's and Children's Hospital in Adelaide, and the Perkins and the University of Western Australia on your side of town as well. So just to give you a bit of an idea of what we actually do with these precious samples, and there's a number of different things. So these samples are consented for research, and they can be used in collaborative ways in any project that has an ethical consent to be able to study them, and we help to facilitate that. But two of the projects that I wanted to touch on are things that we've been doing. And one is really looking at trying to understand what's happening in patients with muscle disease in terms of their immune cells. And these are just some pictures of what muscle looks like when we take a section of the muscle and cut it down the middle. And you can see these little bundles here. They're little fibers, muscle fibers that all run in a nice straight line. And when we see kids or patients with genetic muscle disease, so Duchenne, limb-girdle, and FSHD, what we see is a degradation of this muscle. And a product of that is an increase in immune cells and inflammatory infiltrates that actually impact and drive the disease. And this is really well understood in Duchenne to a degree. And obviously things like steroid treatments, which is the standard clinical care, work quite well. But it's not particularly well understood in other conditions like limb-girdle and FSHD. And steroids actually don't help these patients very well. So trying to understand this in more detail is one of the aspects that we use the blood for. And to do that, I don't want to go into too much of the science or what we're actually doing, but I think this is pretty cool and gives you a bit of an idea. But we can use a really neat machine called a flow cytometer. We can put the patient's blood through this flow cytometer and identify the specific immune cells that are present in that blood. And then during the process of analysis, we can compare these to healthy controls, so kids that don't have a neuromuscular condition, and look at what happens in our patient. And hopefully what you can appreciate is these blue, purple, and pink dots, they cluster differently in the patient. And so this population here is actually a unique cell population that's present in a patient with limb-girdle muscular disease. And that's really when the research happens, is trying to figure out what these cells are doing in these patients. These can be linked to disease pathology and progression. They can also be biomarkers. So biomarkers are circulating markers typically in the blood that can help clinicians and researchers understand the disease progression, but they could also be targets for new treatments and drugs that we just haven't yet identified. And so this is a neat way to be able to understand what's happening in the patient at any one time. And without this equipment and looking at these sort of complex markers, we wouldn't be able to develop these maps of the patient. And so the second part that we can do with these patients, and this is coming to the stem cell component. I realize we probably haven't talked about stem cells yet, but this is sort of the beginnings of the stem cell chat that we want to introduce you to at the moment. And so what we're doing with these cells is we're trying to create what we're calling patient-centric muscle models. So these are models of a patient in a dish. And we use induced pluripotent stem cells, or iPSCs. A bit of a mouthful. But what is an iPSC? You might have heard of stem cells before, and there's actually two types. There's embryonic stem cells, and these are collected, as the name suggests, from embryos. This is not something that's typically used because the material is difficult to obtain, and there's a lot of ethics associated with retrieving embryonic stem cells. But induced pluripotent stem cells are actually grown from blood and skin tissue collected from us as adults in developing, and they can be grown in the lab. And then we use this series of growth factors and chemicals that actually convince them to form back into a stem cell-like state. And this was actually awarded a 2012 Nobel Prize. So realistically, like these types of cells have only been around for about 15 years. And in science, that's a pretty short time. But the great thing about stem cells is that we can reprogram these cells once they've been reprogrammed into almost any tissue of the body now. And we're particularly interested in heart and skeletal muscle. And these stem cells are immortal. They can basically grow forever, which gives us in the lab a source of tissue that can go on forever. So why do we need them? Well, muscle biopsies are quite invasive. Anyone who has gone through one, I've had one. It's not a particularly fun process to go through, and it's really no longer routine. Thankfully, because of genomic testing, we're able to now understand a lot more about genetics from just a simple blood draw. And also the models that we typically use, like mice and other animal models, often don't reflect what's happening in a human setting in a patient. So trying to develop human-specific gene therapies and treatments requires human-specific cells. So how do we use stem cells from patients? So we have what we call a disease modeling pipeline. So we're able to take these stem cells and grow them into muscle cells. It takes about 30 days to produce the progenitor cells that we need to be able to start the process of making muscle, and around another 2 weeks to then get these really nice, we call them striated. So you can see the green fibers here are the muscle fibers running across. They're grown on the bottom of the well of a tissue culture dish in the lab. And so we can do this in monolayer or 2D on a flat culture, and we can also do these in 3 dimensions, and I'll show you some pictures. This is a honours student, Chatu. She's working hard to finish off her honours at the moment. She was working with us this year. But the really great thing about these, which I've sort of mentioned before, is that we're able to go into these cells in a dish and identify the patient mutation and correct it in the dish to create a really well-controlled matched cell line. They're useful in improving disease modeling, so performing functional analyses and also doing drug screening. And we can test, different targeted treatments and disease-specific models in this way. So I'll just show you an image of what these cells look like in addition. So the one thing that muscle is renowned for is producing force and helping us to move. And so you can see these are muscle fibers that are growing in a dish. Now I'll just go back. Now, when they're just sitting in the dish, they're static. They're not moving. They're just going about their own business. But when we stimulate them, we have to put a little electrical stimulation on these cells to encourage them to contract and to get them to move, because that's obviously what their function is. And hopefully you can see these regions here. It's a bit tricky, but these muscle fibers that were grown from our stem cells are able to contract and move in the dish. And so this was really a great first step to say, well, the cells that we're producing, the muscle that we're growing, is doing the function that it should be. So from taking them on this sort of 2D flat layer, we're also able to grow them into these more complex 3D muscles. And you can see this is a muscle group, a bundle of muscle fibers grown up here, and they grow up on these 2 plastic poles. And we can measure, when we stimulate them to contract, we measure the deflection in these poles as a marker of strength and performance. We can also mark these cells with different antibodies to look at how they structurally form. And then we're also able to break these down and really look at how these muscle fibers interact together. So this is a bundle of 5 or 6 fibers. The big blue dots here are nuclei. They contain all the DNA that tell them that they're a muscle cell, and then the different colors. So we're able to use these sort of complex organisms in a dish to be able to understand more about what's going on in a patient and get a better understanding of their disease. Oops, sorry. And then the final thing that I wanted to talk about was one of the aspects of these cells is that it allows us to screen for novel drugs and treatments. So we're able to look, and in this case, we're screening 30,000 different drug compounds, and this is a large number of different disease-modifying compounds that we think could have an impact on the patient's condition. And the only reason why I can do that is because of these stem cells that we can grow large amounts of. We can put them into the cell culture dishes. We use robots to be able to help us screen, image, and change the media and feed them. And so we can take this sort of pipeline where we're looking at 30,000 compounds. We can whittle this down to around 30 to 50 and go into the more complex models. And then out of those that improve like force and function, we're able to take these into another mouse model or another system to be able to look into it more detail. And this work is funded through a really exciting program through a stem cell medicine initiative called Renew, and it's supported by the MCRI drug screening platform. So all of this kind of happens on site from the point that the patient sample comes up to us in the lab, is converted to a stem cell, and can go through this platform. So now that we sort of talked about what we use stem cells for, we really did want to just briefly touch on what's real and what's not in terms of the stem cell space. And Robin, I don't know if you want to maybe unmute and we can sort of chat a little bit more about this. That was sort of my last data slide. But hopefully what I've shown you is that what we're using stem cells for is a really useful tool to model and study disease, to identify new treatments, and to be able to, understand a lot more about what's happening in a patient. There's some really exciting work in the lab that's going on, not in our lab, but in other labs around the world, looking at how we can potentially use these cells as cellular therapy for transplants, but that's really not currently approved, and there's no real treatments available. And we just really want to flag that there are for-profit stem cell clinics out there and around the world that advertise that they can almost treat anything. And I found this image here on the internet just today, and it's from a group called stemcellsindia.com. And they have a whole list of different stem cells or stem cell therapies that can be, or disease that can be cured by stem cell therapies. And really, there is no evidence. There's no current FDA-approved stem cell-based trial. So we just want to make sure that we let you know about this in terms of a word of caution, and to make sure that you contact your clinician to really understand more. And also in Australia, we have a really useful website called Stem Cells Australia, and that's been developed and designed to really help us as researchers and you as patients and advocacy groups to understand and make sense of stem cell science, because there is a lot that comes out there into the media that we really want to make sure that we're clear about what we're doing with stem cells in particular, and where the hope comes from. I don't know if you want to add anything to this, Robin, but
no, I think you've summed it up perfectly. So the work that Pete does in the lab, and when we consent patients for a sample, it's 5 mils of blood that we take from a patient. So that's a teaspoonful of blood. And it continues to astound me that Pete can do so much work from a teaspoonful of blood and do this amazing research. And I would encourage people, if you're thinking about stem cells, that you do have a look at the Stem Cells Australia website, because it's written in lay terms, so it's easy to understand. Sometimes what we see in the clinical setting, when a child or a family member is diagnosed with a neuromuscular condition, other family members are shocked by the diagnosis and upset by the diagnosis and want to do as much as they can, and they don't know how to help a family. So often it's other family members that go on the internet at the time and search for treatments and cures and therapies. And there can be a lot of pressure on families sometimes from friends and colleagues, family members, saying, look, we've read about this, that bottom sample that Pete's shown there is the perfect example. Why don't you try this? The doctors in Melbourne don't know what they're talking about. You can, fix this. If you do read anything, if you do see anything, please discuss it with your clinician. Trust me, if these things worked, we would be doing them first and foremost in our clinics here in Australia. Australia has an amazing healthcare system. But they don't work, and we don't know that there's, what's being done. We don't know that it's safe. We don't know what it is. So please check with your clinicians if you do read about anything. But at the same time, please have hope, because the work that is being done in the research setting is amazing. It really is incredible. I know it doesn't move fast enough for families. And we are well aware of that. The scientists are well aware of that. And that's part of the reason for having this national collaboration and this international collaboration, that we are moving, as quickly and as safely as we possibly can. But I think Pete and I would be happy to answer questions if we can.
Yeah, definitely.
Yeah, does anybody have any questions? If you want to put your hand up, and then we'll unmute you. Just raise your hand if you know how to do that, or just put it in the chat. Yeah, I think it's really important that you just gave a little overview of the hope versus hype, because I think it's a very difficult space, and I think Mike would jump in as well. It's been very difficult, as I've been on this journey with my son now for 17 years, and it's a very difficult space, because we feel in that time we haven't come very far. But as Peter was saying, you know, stem cells are relatively new, and it's not that they're new, but it's our understanding of the science is relatively new in the scientific world. But we have seen the stem cells therapies available overseas for longer than that. And the fear is that potential harm, and, you know, taking advantage of vulnerable families at a time when we're all really looking for information and ways to help our children and families. So the best thing to do, as I said, is to speak to your clinician, get involved with the registry. If we don't know you're there, we can't help you. Reach out to organisations such as Duchenne Australia, and there's other local organisations across Australia as well and internationally that can also support you and give you information. But yeah, if anyone's got questions now specifically for Peter or Robin, we'd love to hear them. Mike, do you have anything?
No, I'll also add, so I mean, I think, you know, so obviously I get a lot of questions around stem cell treatments, because I think there's almost been an evolution in how they're sort of marketed in the recent times, where, you know, typically, as even myself, you know, back 6-7 years ago, when, you know, the diagnosis came, we all searched, you know, Duchenne, QR, whatnot, and you come up with a whole bunch of South American stem cell clinics or Indian stem cell clinics, where now I think we've sort of come into a time where they're sort of even marketed by some very active families and social media. And I have, friends that are very active in these treatments in the States and whatnot. And then it's sort of almost certainly sort of marketed in the way of clinical trials now. And we get these things because, you know, they're crowdfunding for these trials, and they're marketing in the way of we're developing a clinical trial for these stem cell treatments. Now, what we typically say to families, and what the response is, I mean, first, from my perspective, having friends that have pursued these treatments for many years and given up, you know, sold houses and sold cars and really come at quite a big financial cost to pursue these treatments. And realistically, what we've seen is nothing to make me believe that these are anything more than a short-term treatment at best, if that. And our answer to families is, if you're asked to pay to be involved in a program, it's not a clinical trial. You know, so I think that's
absolutely.
And thanks for your perspective on these, Peter and Robin, because I mean that was my main point here, is to get that perspective. But I mean, I think that's the treatment. You know, the way it's sort of evolving now on social media and how we all interact on social media. I think that's a pretty important point to make. Is that that we don't have the evidence, and, you know, typically if it's a clinical trial, you don't have to pay for it. So
yeah, thanks, Michael, that's great. Rick, you've got your hand up.
You might have to unmute.
Oh,
Yeah.
sorry. Yeah.
Okay, I can do that.
Yeah,
sorry, is that better?
Yes,
Yeah.
that's good.
Yeah, no, look, I just wanted to acknowledge both Robin and Peter's work. It's Rick Ferguson here from the Daniel Ferguson LGMD Foundation. I just find the work, as Robin said, with the work you're able to do with just 5 mils of blood, Pete, is just incredible. And from our perspective, it's very exciting. I am wondering if you know anything in regard to something like the Vita Therapeutics, if you've heard of them over in the US, regarding some of their research and regrowing using super cells to regrow muscle. Is that something that's actually being promoted in social media as opposed to being a reality, if you know about it at all?
Yeah, I have seen a few bits and pieces from them. I haven't done a lot of my own sort of research into them, or what they're actually proposing. So I'd have to have a bit more of a look, I think. But, you know, like we were saying, if it's a clinical trial, it will be done under the ethical auspices of a clinical trial, and you know, they are becoming a reality. There is a lot of work that's coming out of the lab, even though it's taking time. So I'll have a bit more of a look, and we can have a bit more of a chat about that, Rick, if you like. And I'm not fully across it all.
Okay,
but,
no, I understand,
yeah.
appreciate that. That would be very interesting, Peter. Thank you for that. And again, acknowledgement to both yourselves and the teams and Robin and the ANMDR for the work that's being done.
Thank you, Rick. Yeah, no, that's Yeah, it's really good. And like you said, there's always so much new stuff coming out. So it's great that we have people like Robin and Peter and their team to talk to. So if anyone wants us to follow up, please email info@Duchenne Australia afterwards, and we can pass on, ask any questions to Peter and Robin. But Sarada's just popped in the chat. She wants to know if it's possible for stem cell therapy in the future to potentially reverse a condition like Duchenne. Peter, you can take that one.
Yeah, sure. So I tend to say it depends. That the hope is that we can catch the condition early enough that the damage that's been done to the muscle can be stopped or slowed. So, I mean, you kind of see and lived experience as the muscle degrades, it's quite difficult to then put new muscle back into an already damaged kind of space. But, you know, there's definitely the hope that the stem cells could provide that kind of improvement. The tricky thing is muscle is the largest organ of our body. When you think about trying to treat a whole system organ like that, it's very difficult. But then also what Robin was saying, you know, if we're able to make incremental improvements to change the quality of life of kids that are developed and progressing with this disease, that's a really great step forward and something that we shouldn't undersell. There's always this thought of a cure, and particularly for young kids, and that's what's happening in SMA. You know, they're treating young babies and children and seeing quite positive effects. SMA and Duchenne are very different conditions, obviously, but that's sort of the poster child at the moment for this kind of condition. So, yeah, that's probably what I would.
That's great.
I would add to that even with spinal muscular atrophy, where we have gene therapy available and we have 2 other very successful treatments available, the treatments are called disease modifying therapies, or DMT. And that really reflects what they do. They modify the disease. So you still have Duchenne, you still have SMA, you still have the disease. But these excellent treatments either help slow down the condition, or they make it not as serious as it would have been in the untreated state. So I think we all, you know, if there was a cure, it would be amazing. But I think the reality is that this work is going to lead to disease modifying therapies. I think that's the aim. I don't know if you would agree with that, Pete. And
yep.
So, you know, we also need to be careful about the terminology that we're using. And then if we can slow a disease down with one treatment, the aim is then if something better comes along further on, you know, that may help improve again. So what these treatments do is they're buying time. You know, they're slowing the progression of the disease down is the aim, and then buying time for further down the track as well.
Maybe I'll just add, so there isn't probably any one cure or one treatment that's going to result in this. So we're looking at combination therapies, and that's also why we're quite interested in the immune system. You know, you have to look at the whole of body approach, which is kind of quite complex, but, you know, the gene therapy treatments on their own may not be as powerful as they would be in combination. And that happens in everyday medicine as well. It's a combination. So I wouldn't be looking for that one silver bullet that's going to be, you know, the answer to all of our questions.
And Peter, just because obviously with stem cells as well and muscle, there's different muscles in the body and they are different. So looking at stem cells in cardiac muscle, for example, and the important impact that even just targeting one part of the Duchenne condition can be a game changer.
Yes.
So, you know, just because I think what we're thinking when we say cure, that's the whole body. So that's physically, you know, skeletal muscle right through to respiratory, through to digestive, to yeah. So do you want to just sort of, yeah, because stem cells are different, aren't they? I mean, I'm not saying
yeah.
they are different.
No, yes, definitely. No, they are very different. So here at the MCRI, we have a dedicated team that are looking at the heart stem cells, and in particular, conditions that relate to cardiac structural abnormalities or genetic defects like Duchenne. So there are groups that are working here on heart-related. So we're able to use those same stem cells that I mentioned before, where we're saying, well, let's look at a skeletal muscle, give them different growth factors and different signals that then tell them to become a heart. And we use very similar platforms to what they do for the heart and skeletal muscle, but they are very different organs. So there are a number of studies that have used stem cells from cardiac stem cells or stem cells that have produced cardiac cells to look at some of these drug screening as protective drugs that might improve heart function in children with Duchenne and some of the other congenital muscle diseases. So I think there's a number of different sort of early stage preclinical models where they have focused on the heart, where they're like, yes, we could probably make an active change in that. And some of those are in early phase or 1 and 2 trials at the moment as well, because the heart is like a dedicated organ there. And the same with respiratory muscles, they're quite different to our postural and support muscles. So thinking about the diaphragm and how we can improve respiratory function would make a massive difference to a lot of the kids that are going through this. So every muscle is different.
Yeah, that's right.
And every child and every adult is different. I mean, that's the other thing as well, which is why clinical trials have selection criteria for the trials. So, this is complicated.
But not impossible. That's why we're doing.
No, that's right.
Yeah, that's right.
Yeah,
that's why we keep at it.
This is what keeps us going. And like, we really appreciate people coming and asking these kind of questions, because it does make us think too. It's kind of like, well, how are we best representing what we're doing? Are we asking the right questions? What could we do differently in the lab that might be able to make a change now? And one of the other benefits that I'll just quickly mention about stem cells and what we're trying to do now is to use stem cells to shorten that time that it takes to get from a lab-based therapy into the clinic. So thinking about ways that we can use stem cells to shorten what might be a 10-year process down to 5. And that's a real driving force between what's happening here at the stem cell medicine program, is to actively use stem cells to shorten this process. So that's quite exciting, I think, from another angle, not only like a therapy themselves, but how they're going to, in the next few years, improve the way that we test, treat, and identify new drugs.
That's actually, Dean has just popped in the chat. That's what he said was, with time being the critical factor here, what does the journey look like from discovery to it being available for treatment?
Yeah, so just as an example, that the one drug study that I mentioned just at the end there, we have a 2-year program where we hope that we'll have drug targets and candidates that will be identified in that time. And then from that, we have a 3-year follow-up grant that is to take those candidates and get them into a phase 1 trial. So that would usually take 10 years and 15 years, but to try and get that down to 5, and would really make a difference in the way that we can progress. And actually, some of the targets that we look for are FDA approved already. So they might be used for diabetes research, or they might have been used for cancer therapies that they didn't quite make it to the end of that trial, because there's a lot of different reasons why drugs don't make it through that pipeline. But maybe we're able to repurpose those drugs to suit our needs. There's a few key examples of that kind of happening.
Yeah, that's excellent. And we've got one other question in the chat, Peter. So if there's a child or an adult that's already on an existing therapy, such as exon skipping or gene therapy, will the stem cell therapies be available to them as well? Is it? If you were talking about combination therapies, is that sort of?
Yeah, so I think there's a real push to be able to, and in a number of different conditions, and FSHD, actually, I was just talking to a few of them, is to make sure that patients and individuals who are on existing clinical trials, when they finish that they are able to stay on that trial and continue to take that drug while they're analysing the data and looking at sort of the outcomes of that phase 3 study. There are then limitations, and Robin kind of mentioned that when there's a clinical trial, there are exclusion and inclusion criteria, and often an exclusion criteria to get onto a first-in-man kind of trial or first-in-person trial is that you aren't on any other treatment programs or experimental medication at the time. But, you know, understanding that the combination therapies are going to be important, we've got, you know, a few different trials happening already here at the Royal Children's Hospital to be able to understand what the implications of some of those combination therapies might be. So I'm not sure if you want to add anything to that, Robin, but...
And many of you will know that just in the Duchenne trials, it depends on the genetic change that you have to be whether or not you may be in or out of that particular trial. And your clinician is always the best person to have that discussion with you. And again, once, you know, these combination therapies perhaps evolve and for clinical trials and potential treatments, your clinician will be the best person to have that discussion with you, and also to be on the registry. So when we update your information annually, you know, we ask, are you on any studies at the moment, any research trials, any therapies? Are you in any clinical trials? So that also helps us know who is on what around Australia. So as different things come up or potentially come up for trials and treatments, we can let your clinicians know and let you know. We haven't really answered your question.
No,
that's an excellent question.
It's a great question.
It's going to be an individual answer for almost every person, I think.
Yeah.
I think you did answer it, to be honest. I think it's just important because when we're talking about that hope side of things, I think people sometimes are hesitant now to go on trials thinking, what's the next thing? If we take this opportunity, or if we don't, you know, are we going to miss out on something else? And do we wait for stem cells and not do the gene therapy trial? So like you said, it's really important to have those conversations early. And we get these questions all the time, and especially for the females with, you know, the manifesting carriers who are excluded from most of the trials, you know, when are they going to have an opportunity? And I think that's the exciting thing with things like stem cells, is they're probably not going to, well, I'm hoping it shouldn't be dependent on sex, for want of a better word, gender. So yeah, I think it's really great that we're having these conversations now. Sorry, Michael, I cut you off there.
No, and I mean, that was, you know, what, if we've got any time left to speak about, is like, so going back to Sarada's question about can it be reversed. So I mean, I think we're seeing now there's a few startup companies and a few companies, I think Satellos is one of them in America, that are marketing. I mean, well, what they're saying is, you know, it's basically what we're starting to label them as restorative therapies, because I mean, they're saying, you know, it's about rebuilding lost muscle and whatnot. I mean, I guess, what does that mean to have a treatment that rebuilds lost muscle, but still have a body that's deficient of dystrophin? So I guess that comes down to having a treatment like a stem cell treatment that obviously, you know, we're talking about engraftment here, that, you know, engrafts into the body and continues to create new stem cells, creating dystrophin, and a treatment that can rebuild muscle. So I think we're a bit in the future, you know, we're looking a bit into the future here, but you know, I think we are moving into that space where there are companies looking towards these restorative therapies that will rebuild lost muscle. But, you know, what's the other side to that? We need dystrophin to maintain these muscles if they can be rebuilt. So I think that's, you know, it'll always come down to a combination treatment. But ultimately, I think in Duchenne, we're going to need, you know, dystrophin, probably full-length dystrophin, to be expressed. And, you know, anything else after that is maybe a bonus in terms of, you know, future science. Yeah.
And maybe just to touch on what you're saying there, Michael, like that interaction with companies and, you know, small, large, you know, that's really important. And what the registry provides, I guess, is that conduit to them, and to make sure that they're coming to us with the best possible therapies, and that we're able to say, yes, this is what we want to do here, and this is how we can get it happening here. So I think, yeah, that interaction and keeping an eye on what's coming out from a company setting is really great. And we can't do this on our own. The research, the academic kind of industry, the clinical side, it's a really multidisciplinary approach, and we need pharma along for this ride. as well. So yeah.
Yeah, and collaboration is key, is exactly what you're saying, isn't it, Peter? And that's the same for organisations. And there's no point in somebody else duplicating if we can build on the work that others are doing. And
well, especially with the limited time and resources that we have, why
would we? Yeah, I think that was what we always used to do, because it was who could win the race. Whereas we know this is a marathon, not a sprint, and it's about getting the team across the line and not just one winner. So I think that's just a little bit of a mind flip that we've all had over the last few years. But
and it's complex. We've been talking about trying to restore dystrophin and return it back to the body, and they're realizing very quickly that while 20 years ago when we identified the gene and thought, oh, we'll have a cure in 10 years, you know, that's not the case. And there are brilliant people working on this. We're scratching at the surface over here, but collaborating with a number of them. So I think, yeah, that connection and making sure that it's an international kind of approach is what really excites me too about what's happening now.
Yeah. And look, we really thank you both for your time this evening, and, you know, to everybody in the community that's online. We will make this available wider as well. But we really thank you for all of your hard work and for, you know, focusing on our community. So not just Duchenne, but the neuromuscular community, because we learn so much from those similar disease groups, and, you know, those that are ahead of us and behind us, you know, we're all sort of in that together. But I think it's important as well that you're so good at being able to give that information at a level that the community can understand, because this is complicated. Duchenne is complicated, and we all feel like we've got to be genetic counselors and neurologists and pharmacists and all of this as we go through the journey. So having, you know, people like yourself and Robin and the team to sort of help us understand is just pivotal. So yeah, we really thank you all for everything that you're doing. And you know, maybe we'll give it till the new year and maybe touch base again and have another webinar, just see how things are going. But yeah,
happy to. No, always happy to talk what research is going on. And if there are questions, yeah, you can email us or go through you guys in Duchenne Australia, whatever you like. You know where we are.
DuchenneAustralia.org. We can direct you to where we can best serve you. So.
Yeah. And join the registry. So we don't want any of the adults online or parents with adults thinking that this isn't something for the adult community. You know, any emerging therapies, we're going to push really hard for access across the whole community and not specific to younger age groups. So yeah, thank you everybody who's online. If you have any questions, just let us know.
Thanks very much, everybody. Much appreciated.
Thanks, Rick. It's lovely to meet you online.
Yeah.
Take care.
Thank you.
I'll be in touch. Yeah.
Thanks, Robin. Thanks, Peter.
No worries. Thanks, Rick.
Stay safe, everybody. Thank you.
Cool.
Bye.
Thank you.
Thanks.